my work sits at the interface of comparative oncology and surgical practice. And so today I'd like to explain how understanding cancer across species, humans and animals, can unlock breakthroughs that can help everyone. Oops. So this is where I work largely at the Royal Dick School of Veterinary Studies, which you can see on the top left there. And you can see on the bottom right, our Hospital for Small Animals, where I'm largely based, particularly inside the cancer centre, where I provide surgical care for our oncological patients and looking at clinical trials as well. I'll just move this off there so you don't get annoyed by that. There we go. So before we go on, I just want to pause for a minute because I am really aware of what we're talking about today is not abstract. We're talking about cancer. It's personal. It's something that I've lived through, both as recently as a parent, but also as a clinician and as a pet owner. And I know that cancer affects our families, our patients, our pets, and our colleagues. So if at any point this feels really heavy, then please feel free to step away. There's no pressure to stay still or be however you think you need to behave about something that is by nature deeply human. And I hope this session is about hope, about connection and shared progress. But I just want to honour how emotionally charged this topic can be for many of us. And so I wanted to share this quote, which is really nice, but also this image of a cairn in Scotland, which is usually seen when you're out walking about the hills. It's a man-made mound of stones used for various purposes. Originally sort of ancient burial monuments, but now as modern summit markers for climbers or navigational aids to sort of find your way around barren land. So it felt really, really appropriate for this topic. So what we're going to talk about today, I'm going to talk about setting the scene, why one medicine matters. I'm going to try and talk about the science and what makes cancer shared. And then a few examples, so particularly in the melanoma space, which is my area of research, I'm going to share some lessons in that space. I want to share an example of some of the clinical research that we've been doing. And then in terms of the bigger picture, in terms of cancer care, a little bit about barriers and breaking barriers, which is really important in the one medicine space. And then some notes about looking ahead as well. So first of all, setting the scene and why one medicine matters. And let's start with something really simple. We all know this, we're not that different. Humans and animals share around 85 to 95% of their genes, and we suffer from many of the same diseases, cancer, arthritis, epilepsy, heart disease. But for most of history, medicines evolved in parallel tracks. We've usually got one for humans and one for animals, which is a little bit further behind. And we built these remarkable silos, each advancing knowledge, but we don't usually look sideways at each other. So one medicine is really about asking us to remove those walls, not about treating humans like animals or animals like humans, but recognizing the shared biology that underpins health and disease and learning about that together. And beyond biology, it's really about shared compassion. The bond between people and animals is already really deeply emotional, and one medicine really makes it scientific as well, turning love and care into better health for all species. So what do we mean by one medicine? It's the idea that human and veterinary medicine are not separate worlds, but they're two sides of the same coin. And when we study disease in one species, we can see insights that directly help the other. And just some examples, dogs develop epilepsy that can look and behave like human epilepsy. So studying that can be really, really helpful in helping neurologists diagnose and manage seizures in people. In cardiology, we've discovered that the same gene mutation causing heart disease in cats and humans is present, and that can lead to better health and screening and treatment strategies. I think a really powerful example is large breed dogs with osteosarcoma, which is a bone cancer. And this is a bone cancer that's seen in children as well. So we can use examples where understanding the disease in dogs can help refine treatment in humans as well. Orthopedic and regenerative work with stem cell therapy for canine arthritis has helped shape human joint repair techniques. And even amongst mental health, that connects us as well. There is studies of PTSD in working dogs and their handlers. We can see the same sort of stress pathways in response to therapy. And an example would be that the dogs that we used in 9/11, they became very depressed by not finding living people. And so they had to take them away and do a body of work with them where they mocked up a rescue situation to allow the dogs to find live individuals, because they found working in those environments so distressing. So there is a massive link across a huge variety of disciplines. And cancer is the perfect one medicine example, because it affects all mammals and behaves similarly at the cellular level and challenges us emotionally in the same way. At its core, cancer is the same biological story repeated across species. Basically, what it is cells that they forget when to stop dividing, essentially, is cancer. And whether those cells belong to a person, a dog, a cat, a horse, a cow, a chicken, a fish, a fly, they all follow the same basic rules of growth and repair until something in that cell goes wrong and the cell develops in an uncontrolled manner. And we see that the same genes are often to blame. And I've listed some here. So the first one, BRAF, we see that often in melanoma. TP53 is often called a guardian of the genome because it fails in tumours across many species. And KIT mutations as well, seen in dogs and people, for example, in gastrointestinal tumours. So once you've got this change, then the process of the development and spread of cancer is strikingly similar as well. The tumour starts to grow its own blood supply in order to allow it to survive. It hides from the immune system and then it spreads. And even when we choose to treat the cancers, then we have similar patterns of resistance and response in humans and across animals. And it's really interesting because some animals are actually better at avoiding cancer. And there's some work ongoing, particularly in whales. They're very, very large mammals. So you think because they have so many cells, you would have a higher risk of cancer because each cell has the same risk of being damaged. So if you have loads of cells, the chances of developing a cancer are higher. But actually, whales do not. And so there's a lot of work ongoing to try and understand why that happens, and we can learn something from them. So cancer is a really fertile ground for one medicine because nature has done an experiment for us, and we just need to look at it and understand it. So why don't we do that already? Why are we not brilliant at it if the science is so clearly shared? And there's some very real barriers, not only in biology, but in the systems that we've built around it. And sometimes we're not taught the same when we go to medical school or vet school. We're taught in silos. We don't often look across at our medical or veterinary colleagues. Our research models are still rooted in the lab, and we're not going to talk about the ethics of that because that's a whole different topic, and I understand and recognize that. But importantly, the lab models don't have very good translation into therapies for humans or veterinary species. So only about 5% of therapies tested in animal lab trials ultimately gain regulatory approval for use in humans, because the mouse or the rat or the fish, it's not a very good model because that disease has been engineered in that animal to represent it. And part of our work is trying to say, if we look at our veterinary patients, those diseases occur naturally in animals that live in the same environments as us. They're better models. We can use our veterinary species to inform human disease. We can use our humans to inform veterinary disease and try to minimize the use of lab models in that space. There's ethics and regulatory frameworks. They often operate within the veterinary space and the human space completely differently, with two different languages of consent, governance, and oversight. So sometimes it can take a long time to align the approvals. I think one of the major problems is funding, because veterinary oncology receives less than 5% of global cancer research funding, but it's one of the most richest translational opportunities. And usually funding resources are very siloed and they're very geared toward the human disease. So therefore, trying to do work in the veterinary space is really, really challenging. And that's where the Humanimal Trust is unique in recognizing that collaboration and funding projects that allow human and veterinary work to go hand in hand. Treatment access is another gap. Many cutting-edge human therapies take years or decades to reach veterinary patients. And of course, there's cost implications. We don't have an NHS for animals, so there's cost implications associated with that. So that means that even though those same patients helped us to understand the disease, it often is a little bit longer before they can benefit from the treatment. And then finally, there's culture barriers in terms of data being stored in systems that doesn't speak across the different species, professionals trained in parallel but not together. And so the end result is we have a world where we've got these two communities, each really full of compassion and innovation, working towards the same goal, but they don't do that in step. And so One Medicine really challenges us to fix that, to try and redesign the structure so every discovery moves in the same direction. So if we look at animals in cancer research and humans in cancer research with the aim to bi-directionally help everyone, really, when I speak to medics, human medics, they're often really surprised that dogs and cats and horses get cancer. And that's a bit of a shame, because when we talk about comparative oncology, we're not talking about a metaphor. These cancers are real, and the dogs genuinely develop them in the same biological context that we care about. They live in our homes, they eat our food, they live in our environment. And increasingly, our dogs and cats are receiving the same healthcare systems as we see in humans. So there's some examples of shared cancers here. I've already talked about osteosarcoma, which is a bone cancer. That's a very good model of a children's cancer. Lymphoma, which is a blood sample that blood cancer that my son was diagnosed with earlier this year. We see the same cancer in dogs and in cats. Breast cancer, really, really common cancer, particularly within the UK. Very, very common cancer. We see the same cancer in dogs. Bladder cancer, same cancer we see mechanistically in dogs. And then my area of research, which you can see down here, which is oral melanoma, which is a cancer of the mouth, the most common cancer in dogs, but a rare human equivalent with a survival of about 15% over 5 years. So we've got this amazing platform in our veterinary species that is enormously underused because people just don't know about it. So our patients develop the same cancer, but we treat them in the same way as well. And that gives us a really rich platform. So we steal all of the therapies from human medicine, and we have an opportunity to give back data to try and align treatments between human and veterinary medicine. So our platforms that will treat surgery is the mainstay treatment for most cancers, and that's my role. I'm a surgeon, so I will provide surgical care for our oncological patients. Chemotherapy provided to our dogs and cats, same protocols, same drugs. Radiation therapy, this is our radiation unit at the Edinburgh. It's exactly the same as the unit they use in the NHS. We can use immunotherapy, we can use palliative care, and then of course end of life care within our veterinary species as well. And my research is related to melanoma, oral melanoma, which is really, really good, in my opinion, gift to us to allow us to study the disease to provide benefits to both species. And I'm going to show you how we try to do that. So oral melanoma is absolutely devastating. It's the most common oral tumour in the dog. I think within our hospital, we see about 40 or 50 patients a year. We've just started a clinical trial that we've opened up via a single Facebook page. I'm now receiving between 15 and 20 emails a day from people whose pets have received a recent diagnosis. The survival is not great. The spread is very, very common, and the therapies that we have aren't great. If we look at people, the disease is exactly the same. The tumours develop in the mouth, in the nose, in the sinuses, and there's a similarly poor response to therapy. And as I said, most patients die within 5 years of this treatment. The dog is great because the dog, melanoma was the first tumour that the immunotherapy, so that means drugs that target the immune system, and that's very, very valuable. Melanoma was the first tumour that canine immunotherapy was developed for, which is super. So we have this opportunity where we have two diseases in two species, exactly the same. They behave exactly the same. Let's explore those further. The next two slides are images of individuals with tumours and surgical images. So if that is going to be too challenging for you, then I'll just look away for the next two slides, and then I will tell you when they are finished. So the first thing I'm going to share with you is a picture of a human with an oral melanoma and a dog with an oral melanoma, and I'm going to do that now. So here what we're looking at is, on the left are people and on the right are dogs. And what we can see is we can see an oral melanoma within this part of the mouth within a human, and within the same part of the mouth in a dog. This is a CT scan, which is a 3D X-ray. And what it's showing you is the tumour itself within this individual is destroying the bone of the upper jaw. And we see the same pattern within the dog. We can see that this area of bone is being destroyed by the tumour itself. This is a lab sample of the tumour, and what we can see is the same types of cells in the humans as we see within the dogs. So these tumours behave very, very similar biologically. I'm going to show you, the next slide is some surgical pictures, intraoperative pictures. So if that's not for you, then look away. I'm going to do that now. This is a patient who has oral melanoma, and you can see this here in his mouth. And the treatment of choice currently is wide surgical resection. So for this individual, this meant removal, here's his tumour. This meant removal of the whole part of this lower jaw for him. And functionally, dogs, you can remove large portions of the top and the bottom jaw. They do very, very well. Cosmetically, the result is okay. This is where the fur has been clipped. So this is why this looks a little bit weird. It's clipped in order to prepare the surgical site and make sure that it's sterile, and that will grow back. And you can see him here some months after his surgery. But the most common cause of death in our dogs and in our humans is spread. So we can do this surgery, but the question is, does it actually affect the individual, the end result, because they die of disease spread rather than from local recurrence? So what we wanted to do is to have a look at the human and the dog tumours. And when we look inside a tumour, we can read its genetic personality, which is the pattern of the genes that are switched on and switched off. And within melanomas, we have sort of hot tumours, which you can see here on the right. And these are tumours where the immune system, the infection fighting cells, flood into the tumour to try and get rid of it. The body recognizes that this shouldn't be there and it tries to fight back. The other type of tumour you can get is a cold tumour, meaning that the immune system doesn't recognize it when it's there. And so what we did is to have a look and see in humans and dogs, can we see this in oral melanoma for the first time? And this is really important because if you have a hot tumour, you can respond to a type of therapy that will influence your immune system. If you have a cold tumour, you need a different type of therapy. So what we did is we enrolled some humans and some dogs with oral melanoma who've had surgery previously. And when you have surgery, your tumour is removed and it is fixed inside a wax block here. And a little sliver is taken off, which you can see here, and that allows the laboratory to look at the cells and provide you with a diagnosis. But these wax blocks go into storage, and they're in storage for usually at least 5 years. If you're a person, probably forever. So we have this treasure trove of wax blocks from humans and from dogs. So we interrogated that, and what you can do is you can cut off a bit of tumour and you can extract the DNA from that old tumour that may be a year old or it may be 20 years old, and we can sequence that and see what we find. You get an awful lot of data. So within our institution, we have a tremendous sort of mathematics department that can analyze that. And what they found is humans and dogs have exactly the same 2 types of melanomas. They have a hot type and they have a cold type. And that's important, as I said, because the 2 treatment types should be very, very different for these individuals, depending on which tumour type you have. And that's not used in veterinary medicine at all, and it's not used commonly in human medicine. Now, it's not very useful if you have to follow this whole pipeline for every individual, because this is 2 years worth of work to get to this point. So what we found is that you can identify 41 genes that will allow us to tell you whether your tumour belongs in the hot category or in the cold category, and that will inform decision making regardless of whether you are a dog or a human. It's exactly the same 41 genes. So this part of the talk, we can conclude that archived tumours can be used during research. Dogs and humans have the same 2 subtypes, and we've got a 41 gene signature. So I wanted to share with you just really quickly how that might inform what we've been doing and tell you about our clinical trial. This is in collaboration with a group called Cubiotics, which is an Australian drug company, and it's using some berries that were discovered in the rainforest in Queensland. And you inject the drug that's been extracted from the berries into the tumour itself, and it selects for the cancer cells. And what it does, you inject into the cancer itself, and the cancer sort of dies and falls off. And I'll show you some examples. But also what it does is by killing the cancer cells, the immune system recognizes the cancer cells and mounts a response, and that allows any spread to be treated as well. I'll show you some examples of work that Qbiotics has done. Here's a canine patient with a tumor. And what you see is when you inject the drug into the tumor becomes sort of dies, and then it falls off and heals very, very quickly. I don't work with horses, but I wanted to share some other species because I know my work is around dog, and this is dog heavy, but we're talking about one medicine. So we see the same thing with horses as well, around the eye area in different tumor type and around the vulva in a melanoma. In humans, let's use that information in humans. And what we can see, this is skin cancer, melanoma. We can inject the drug into the melanoma and the tumor itself will become damaged and will fall off and will heal within 29 days. So we started off with our dogs and our horses. We went to humans. Let's go back to dogs now. And we're feeling a little bit safer. So maybe we can be a little bit braver in terms of our drugs. So this is our first patient. This is a Labrador. He's 11 years old and he has a melanoma that has failed surgery and failed current therapy. We can provide the drug into the tumor itself and we see the same sort of pattern. It becomes red and then it falls off. Because of the immune recruitment, you can see the side of his face is a little bit puffy, but nothing too dramatic. And then we see within 24 days, that site is healed really nicely. And he hasn't had any of his jaw removed, as we saw with the Rottweiler previously. So my take home here is we can use the same drug across species to learn. We need to be a little bit careful, and this is why vets need to be involved, because sometimes the metabolism of cats is unusual, and sometimes there's breed-specific nuances that we need to be aware of. So just to mention that briefly. If we look at the bigger picture, what else can we learn? The bigger picture really isn't just about the science that connects us, it's the system, the way we deliver care, the ethics, and even the environmental impact as well. We've got to be careful about that. So in human and veterinary hospitals, cancer care is really resource intensive. So within human spaces, they're often looking at ways to maintain sustainability and reduce environmental impact. And we've led some work in that space too. Veterinary innovations can inform human care as well. And a great example would be the limb sparing techniques, really, really pioneering in veterinary medicine that have then gone back into the veterinary space. And those individuals might commonly be patients with osteosarcoma, bone cancer that we've talked about. The connection extends to ethics too. We face the same dilemmas in both worlds, really. How far to treat, when to prioritize quality of life, and how to support families and owners. And of course, the emotional toil is shared on clinicians, on caregivers, and on the animals and the humans that they serve. So one medicine isn't just about molecular parallels, but about shared responsibility, because true progress in oncology is going to be caring really for the whole ecosystem that surrounds it. So I talked to you briefly about the 41 gene signature that we have developed and that Humanimal Trust is funding discovery of and development of. And this platform, similar platform, does exist in human medicine. So within breast cancer, prostate cancer, eye cancer, skin cancer, you can have a gene signature, so many genes, 15, 21, however many, that you can test for, and that will help you in your decision making. And I was at a conference last week, a cancer conference, and I was looking at all the information that was available for human medicine. What does this actually mean? It's really important that if you are going to have a test, a cancer-related test, that you are appropriately counseled for that test. And during my time at Edinburgh's Sick Kids Hospital with my son, I saw firsthand how research isn't about data, it's about people's stories. This is the oncology clinic in the Children's Hospital in Edinburgh, and you can see it's designed with children in mind by children. So it's sitting beside families in oncology clinics, be that veterinary or human, to help them understand how does the treatment feel? What is it that you're looking for? How is the information shared and how is hope communicated? And this is captured in something called PPIE, which is Patients, Public Involvement and Engagement. And we have a big facility within the University of Edinburgh, within the human space, that looks at this, that draws in patients to design the hospitals, to design the trials, to design the drugs. And so we're stealing from them, and in collaboration with them, we're building PPIE directly into our One Medicine cancer work through a structured program of interviews and engagements supported by our research grant from the Human Animal Trust and also by a team of veterinary undergraduates where we're able to train the next generation. So we're speaking to scientists, clinicians, caregivers, to understand where collaboration feels easiest, where it's blocked, what is it that you're looking for in a treatment for your canine pet. And these conversations are as valuable as any data set because they help us translate that gene signature work, which is the highly technical molecular discovery, into something that's guided by people and animals at its very, very center. So just a quick note, because I know that I've run out of time, sorry, as to how that do we make this vision real, not just an idea, but a working system. So hopefully I've tried to collaborate that we need shared infrastructures, biobanks, that I've talked to you about, the wax blocks, data systems, information that can move safely between veterinary and human patients, aligned ethical frameworks that we're trying to work with, and we're building and making good progress with that in Edinburgh. Collaborative funding. At the moment, many great ideas fall between the cracks between medical and veterinary funders. So it would be really great if each major grant body had a joint One Medicine review panel evaluating projects that serve both sides. In order for that to be realized, I think people need to understand that animals get cancer and they're not that different or that special from us humans. They're exactly the same. Training and education, imagining a generation of clinicians that are fluent in both language, veterinary speak and human speak, is very, very exciting. And joint communication. And I think these are all practical, achievable steps. They're already steps that are starting to take place in Edinburgh and other places around the UK. And the walls are cracking open. We just need to keep pushing. So I hope that I have shared with you today that One Medicine isn't a theory, it's already happening. And every time a vet or a doctor shares data, every time a researcher looks across species for answers, or a family lets their animal experience inform the next clinical trial, then that's One Medicine in action. And what we're really talking about is a shift in mindset, seeing cancer and care as something that we share, really. Because I guess because when you stand at the bedside, whether it's in Sick Kids Hospital in Edinburgh or whether it's in a veterinary hospital at the Riddle Swan Cancer Centre, the feeling that everyone has is the same. It's that of love, it's that of fear, it's that of hope. And biology doesn't really care which species we're in. So it's talking about bi-directional benefit in people. What can we gift in the cancers that we see in our dogs more commonly than we see in people, like melanoma? And in dogs, what can we gift our dogs? And that is the answer, is going to be access to treatments that we enjoy as people, but are not yet available to our veterinary species. And the small project that I've shared with you, that's taken us 2 years. It involves a lot of people. The most important people are, of course, our patients and their families, most of who aren't with us anymore. But of course, they consent to this in the midst of real illness, and so none of this work would exist without them. And then I'm happy to take any questions, and I've just shared some random photos of important people and places and things that are important to me this year. So I'd be super happy to take any questions. And if you are interested in taking part in the PPIE study, then my email's at the bottom, and I'd love to hear from you.
Thank you, Kelly. That was really excellent talk. Really brought together the kind of one medicine and the kind of need for the bi-directional conversations between all the professional groups. I'm going to open up. Has anybody got any? We haven't got any in the Q&A. Has anybody got any questions for Kelly that they would like to ask? You can use the raise hand. Yeah, Eva.
Hi, yeah, great presentation. Just slightly is probably a bit of a personal question, Kelly. What would you say was your eureka moment in terms of recognizing that one medicine was actually a really helpful way forward?
I think it's got to be the first patient who we treated with the drug that I shared with you. And he has been very valuable because he has informed the treatment of at least one human who was on a compassionate care, patient care pathway. So he has informed that individual, which is just something really special. And I think when you can link everyone together in that way, then I think that's really incredibly valuable, that we can have this bi-directional conversation, that we can steal things from human medicine and medicine can steal things from us, and gifting in a circular loop, I think is really important.
Thank you.
Kerry Beacon.
Hi there, thank you for the CPD, it was wonderful. I suppose, I'm sorry if this was raised earlier, I came in about, I think, 10 minutes past. I'm a vet, and is there, in regards to your studies or anything upcoming, is that something you would like circulated through the veterinary community? Because I think a lot of people won't be aware that that is an option and how their patients actually can contribute to this research and benefit from it.
Yeah, that's super valuable comment. So if you as a human have a condition and you want to explore whether there are clinical trials, there's a very well developed system where you can search through all the clinical trials according to country, condition, whatever it is that you want to search for. And that just doesn't exist within veterinary medicine. It's not as well, I guess, developed is the right word. So the clinical trials that we have, or that this melanoma has just been advertised via, there's a large Facebook page called Vet Voices that's been advertised on, but it's very much word of mouth, and the veterinary community is very small, but it's not small enough that we can just rely on social media platforms or word of mouth or engagement. Our caregivers are very motivated, and many of the people who have contacted me, they've found me themselves. So part of the PPIE work is going to be chatting to vets and chatting to caregivers and saying, how do you expect to find out about these studies? You know, is it okay to find out about it via a Facebook page? Or do you expect your vet to know? Or do you expect there to be a platform where you can go onto and you can discover all these clinical trials that are available? So thank you. It's really important.
Thank you for that. I haven't seen it on Vet Voices, but obviously there are a lot of different comments every day on there, so it may be worth me just giving a little nudge on there again. Just
Yes,
from
please do.
visibility.
Yes, please do. I'm very happy if you want to share any content, then I'm very happy to do so. None of it's sort of restricted in any way.
Super. Thank you, Kelly.
Of course.
Fabulous. Anna Harrison has a question.
Thank you. That was brilliant. Thank you so much. I can't actually see the Q&A. I can see the chat, but it's all greyed
Oh,
out. So
sorry.
I will ask, how did that berry get found? Was it just a chance thing, or
It's
and
a
can we
really
go out
good,
looking for more berries, please?
yeah, it's a really good question. So I won't pretend to entirely understand because I'm not a botanist, but the company, the CEO of the company, he is called Paul, and he is a botanist. Where is the picture? Let me try and find it. Hold on. Where have you gone? Here we are. So he's a botanist by trade, and he lives in Queensland. And what I understand happens is they go on a sort of search expedition around the rainforest with the permission of the Australian government. There's lots of licenses and consents, as you would expect, of course. And he can select, leaves or berries or flowers or bits of plant that he thinks might have treatment potential. And then they take them back to the lab and they will then analyze the different extractions that they can get from the berry, and try and do some computational modeling to determine whether that is going to be useful. From this berry, they've also discovered a wound treatment drug as well, which is very exciting. What is the problem with this berry, even though, it's absolutely fantastic, is it only grows in this area in Queensland, in Australia. So what the company has now is like a farm, I guess, in a small portion of Queensland where they grow the berries, they grow the plants, kind of like a vineyard, I guess it looks like. But you can Google them, and they have, they're very open, very collaborative company. And they have a website where you can have a look at all of their discoveries.
Thank you very much. That's amazing.
Yep. And a question from Scott McRae.
Hi, Kelly. Thank you. That was really, really interesting. Clearly, given the gene expression similarities between the two species, this was a fairly obvious target. But I have to ask, are there any other conditions that you have high up on the priority list that you think might benefit from the same approach?
You're talking about the berries?
No, I'm talking about the oral melanoma.
Generally, yes. Yeah, so my area of research is in oral melanoma, but what I am hoping to sort of share is to kind of forge the ground and develop the pipeline that anyone can then follow. So all of this data is published online. You can use the pathway, you can download the gene signatures, and you can analyze it yourself if you so wish, but trying to develop the pipeline so that anyone can come in alongside and follow behind with osteosarcoma, for example. Squamous cell carcinoma might be another example that you see across species. We are developing a unit within Edinburgh called OncoEd, which is a genuine platform between the medical school and the veterinary school and the schools of social sciences, economics, biology, chemistry, to try and facilitate research. If you have a question but you don't know how to answer it, come to us and we will connect you with all of the people. And there's lots of very clever people out there, and you don't need to know the answer, you need to know the question. And then once you've got a question, someone will be able to help you find the answer. So I think there's plenty of other tumours that can benefit from this approach. The limiting factors, in my opinion, is funding, because a lot of the big funding bodies don't get it that companion animals can get cancer, and they don't see that it's relevant. Whereas a mouse, it's easy for them to understand that this is a model, and so we can fund the mouse. We're really good at curing cancer in the mouse, but that's not super helpful for anyone, really, I don't think. I don't know if that's answered your question or if that's just me rambling on a tangent.
Really valuable insight and really encouraging, and I think it gets to exactly what Joe and colleagues are trying to do in terms of promoting that as well. So thank you.
Thank you.
Okay. If anybody, is there any other questions, raise your hand now. I know we're a little bit over time, but this has been absolutely fantastic. Kelly, thank you. I know Joe wants to come in and just say a few words, so I'll hand over to Joe.
Yeah, thank you. Thank you, Kelly. Primarily, thank you to everyone for attending and listening to this. And I'm sure everyone here agrees, Kelly, that was an absolutely superb presentation, one that resonates with professionals, human and animal medical professionals, but also our lay audience and supporters, I'm sure, as well, who have an interest in this. I've certainly learned so much. I want to say thank you, Kelly, not only for the studies that you're doing, but also sharing parts of your personal journey with this as well. I think that really moved me, certainly. But yeah, thank you. I love you said you said a lot of things that really resonated with me, but you said one medicine is not just a theory, it's already happening. And it is, but in a small way. And we really need to get it out there. We really need to get that evidence out there. And your study is going to help us do that. So thank you very, very much.
Thank you very much. And thank you everyone for coming and listening. I'm very happy to communicate with people. If anyone wants further information on the trial, I'm very, very happy to share, or anyone wants to join in the PPIE. Then there's my email. You're very welcome to contact me.
Thank you.